Okay, so I thought maybe it would be a little bit more helpful to do a recorded review type thing. So we're gonna be moving pretty quickly through this, but obviously, with the recording, feel free to pause, fast forward, and just go to the parts that you think you need the most help on. So we're gonna look at overall the project framework, plasmids and transformations, PCR, and restriction enzymes and ligations. So I think that this is the rough roadmap of what we're going to be going through. So we're going to start by just looking at the big picture. So generally speaking, I think Dr. Smith and Dr. Lowe really love to test on like what is overall going on. Like I think in molecular biology, it's really easy to get tied up in the nitty gritty, but it's also very important to have a 10,000-foot view, right? So we're going to be looking at the ADH3 reporter and then IXR1 CRISPR. And for IXR1 CRISPR, that's obviously something that comes later. It's good to have an understanding of what it is right now. But anything past like the general understanding of I think what we're gonna go over is not really necessary, given that we haven't really done any experiments with them yet. We're gonna talk about plasmids and transformations. We're gonna talk about PCR and primer design, restriction enzymes and ligations, and then we're gonna do some calculation drills. I might move the calculations to a different video, but we'll see. So it's stay tuned. Okay, big picture. So what is this class actually doing and why? So we're studying ADH3, which is alcohol dehydrogenase 3. It's an enzyme in S. cerevisiae, and we're doing this through two different approaches. So the two tools involved are, firstly, the ADH3 reporter plasmid, which is something that we've been working on pretty intimately the past few weeks. And then later on, we're going to be doing CRISPR mutated yeast. So the point of the first tool is to isolate the ADH3 promoter by PCR from the yeast genomic DNA, and then you can ligate it upstream of the beta-gal in our PLG670Z. The ADH3 transcription will drive beta-gal expression, and the reason why we're doing this basically is the same promoter that's driving ADH3 is also driving beta-gal, but beta-gal is a lot easier to assay and measure. So one of the important assumptions here is that this regulation is transcriptional, and I think it's important to make that clarification too, like why specifically we're looking at what we are looking at. Okay, so the other tool that we're using is knocking out IXR1 in yeast using CRISPR. So IXR1 is something that's suspected to regulate ADH3, and the other approach that we do in this class is knocking that out. But again, you don't really have to worry too much about what we're doing with IXR1. You should just know, like, very generally, like, we're using CRISPR to introduce a frame shift in this transition factor in S. cerevisiae. Cool. So with that, let's move on to some of the more specifics. So why are we using a fusion protein? So if we look at what we're cloning, we could just put the ADH3 promoter directly upstream of the beta-gal start codon, but we don't. And the reason why we're doing this is we include the ADH3 promoter plus the first 17 amino acids of the ADH3 open reading frame, which will fuse to beta-gal. So the reason why we don't want to start with beta-gal codon directly is we're using two different, we're using different organisms, right? And, we talk a lot about how DNA is conserved and the genetic code works everywhere here and there, but there's some important nuance to that, right? Because fundamentally speaking, in a prokaryote and a eukaryote, the way that we read sequences is slightly different, right? And that tends to have a lot to do with the translational initiation and the efficiency as a whole, right? So translation requires specific context around the start ATG. So beta-gal is an E. coli gene, so its ATG sits in something called a Shine-Dalgarno context, which is just completely wrong for yeast. So by keeping the ADH3 ATG and the bases around it, we preserve the Cossacks context that the yeast ribosomes recognize. So basically, it's just the fact that if we put something that's prokaryotic, E. coli, into something that's eukaryotic, yeast, while the DNA and the genes and like a lot of stuff is conserved, the actual reading of the sequence and the transcription and translation of it isn't really working the same way. Like you don't have a Shine-Dalgarno sequence to recognize where to start and properly do your translation versus in eukaryotes, you need a Cossack sequence. So all this is ensuring is that we've added some extra flanking base pairs to make sure that when we move from a prokaryote to eukaryote, the way that the eukaryote recognizes the sequence is it's compatible with what we've moved and shuffled over from the prokaryote. So I think that's a pretty important distinction. Okay, so PLG670Z is a shuttle vector. So it contains elements that work in both E. coli and S. cerevisiae. So this is really important, right? Like, you think, like, why are there so many different things involved, right? And that's because it has to work in 2 different organisms, right? So it needs to be replicated. These constructs need to replicate, and they need to be selectable in 2 different organisms, right? So it carries one set of elements that's going to work with E. coli. So that's the origin of replication, antibiotic resistance, and then a separate set that works in yeast, which is the 2-mu DNA origin and the URA3. So I think a pretty good question is to ask about blue and white screening in this context. But I think maybe don't worry for it now. We'll get to that in a second. But I think just big picture here, the reason why it needs both elements is because it's used in both species. Okay, awesome. So let's get into plasmids and transformations. So this involves vectors, blue-white screening, getting DNA into cells, etc. So it's important to note that there's 4 types of vectors that we kind of discuss in molecular biology. There's the plasmid. It's the classic workhorse. It's basically the thing that's going to be used for almost everything in molecular biology. And throughout this class, we're just using a plasmid, right? So the whole point is it's very favored for when you have an insert size limit under 10 kilobases. Once you get higher than 10 kilobases, it doesn't really work that well. That's when you have to start using stuff like lambda phages, M13 phages, things called cosmids, artificial chromosomes. And I'm ripping through these pretty quickly, but I think it would be good to have a general understanding of what are the different types of vectors, what are their size limits, and, you know, like roughly where would you use each type of vector. But again, I just wanted to emphasize, for the purposes of this class, we're just using a plasmid as our vector. Yeah. So the general anatomy of a plasmid is as follows. They have origin replication, a selectable marker. They have MCS, and a lot of times reporters and promoters. I think it's important to note, though, that if you ask, like, what are the minimum number of elements that a plasmid needs to be useful, it's really only the origin of replication and the selectable marker, technically. The MCS makes it really, really convenient, but it's not strictly required. Some things about the MCS is, again, like, you know, it's not strictly required because this is like an engineered thing, right? When we're designing plasmids synthetically, we are building them, right? So it's something that we use to introduce multiple restriction sites. So each site will only appear once in the plasmid, so the enzyme will cut in the exact right one place. So let's look into a little bit more about why we use the lab strain that we use. So at the beginning, we prepared comp cells using a strain called DH5 alpha. So these are very heavily engineered competent cells that are designed to be really good at receiving and propagating recombinant DNA plasmids. So there's a lot of different mutations. I don't think you're really on the hook to know, like, the ins and outs of all of them. I think I would really understand the first mutation because I think that's the one that's really very important. I think if I would have to split them up into categories, right? Basically, like, the first mutation, the LAC-Z delta M15, is one category in itself, and it's kind of the thing that actually allows you to make functional enzymes. So basically what's happening is you have a host that's carrying a non-functional truncated beta galactose called the omega fragment. The plasmid carries the alpha fragment, and alone neither works. Together they can complement each other to make a functional enzyme. The rest of the mutations, are kind of just all about keeping the plasmid intact once it's inside. So it's like eliminating all the cell's different defenses against foreign DNA, right? So End A1 inactivates an endonuclease, so it doesn't degrade the plasmid. Rec A1 disables homologous recombination, which keeps the plasmid stable, etc., etc. So I think that, again, in class, you guys covered blue and white screening a lot. So I think I think we should go over it because I think it's pretty important as a general technique in biology. I want to emphasize, though, that we didn't actually use blue and white screening for our experiments. We we used BAM high and SAL1 directional cloning instead. But this is a very common, again, thing in molecular biology. So I think it's pretty good to understand what's going on and what are the different parts of it, right? So the overall logic is you have 3 different outcomes. Blue is bad, okay? If you get a blue colony, that's not what you want. You want white colonies, and then no colony is just no colony, right? So blue involves having an empty plasmid. You have an alpha fragment that's intact, plus the omega from the host, which leads to a functional beta galactose. It cleaves the X-gal, and it ends up being blue. White means that you have an insert that disrupted the alpha fragment, open reading frame, inside the multiple cloning site. You have no alpha fragment, therefore no enzyme, therefore colorless. So this is really, really critical. So the whole point is your insert is actually like within the alpha fragment, okay? So with that, like the thing that you're trying to insert when you like make this recombinant DNA, like the DNA that you're trying to put in to the base DNA, is sitting inside the MCS within the alpha fragment. So all you have to know is if you get a successful insertion, that means you're going to disrupt that alpha fragment because of the fact that the open reading frame is inside the multiple cloning site. And because of that, there's no alpha omega interaction, which means that you don't get functional beta galactose sidase, and then you don't get a cleaving of X-gal, therefore you don't get a blue color. So again, it's really important to emphasize that the white colonies are your recombinants. So yeah, I think that that kind of basically covers what's important. I guess the other thing to kind of note, which I kind of touched upon in the previous slide, is the host strain side. So the LAC-Z delta M15 is the thing that contributes to the omega. That's the thing that you get like the omega from the host for. And the plasmid side, the LAC-Z alpha is coded onto that plasmid. So again, just to emphasize, just know that the MCS sits inside the alpha fragment, which is why an insert would disrupt the function. Therefore, if you get blue, you did something wrong, it didn't work. If you got white, that means it was correctly done and you have your recombinants. Cool. So let's talk about transformations. So transformations essentially describe how you get the DNA into the cells, right? Because we know that DNA is the instructions under which the cells will do all this different, I don't know, they'll perform all these different functions, right? But we need to get the instructions to the cells. So there's 2 different ways that we can do this. There's chemical transformation and electric transformation. So for the purposes of this class, generally we do chemical transformation. Electroporation is actually a much better method in terms of its efficiency. It has a 2 to 4-fold efficiency over chemical. But, you know, for the purposes of this class, we don't really need a super high efficiency, which is why we're using a chemical transformation. And generally speaking, like the process of doing a chemical transformation, preparing like chemical competent cells versus electrical competent cells it's just a lot simpler. And, you know, again, it's for the purposes of this class, it's sufficient and it works, right? So for a chemical transformation, you have calcium ions that bridge the negatively charged stuff. So that's the LPS and DNA, right? So with that, the procedure is, right, you do the shock and then you put them on ice, right? And that cold incubation will let the DNA actually bind. Then you do a brief 42 degrees Celsius shock to push the DNA across, and then you plate, right? So electroporation is pretty similar, you know, like chemistry, like heat and electricity are both forms of energy. They're both ways to kind of open up the cells. Again, electroporation is technically more efficient, but it also involves using a lot of pretty expensive equipment, which means you can also end up spending a lot, and you can also fry the cells a little bit more easily. I think in general, chemical transformations, although they are technically a lower percent efficiency, are actually more reliable for non-specialized use cases. So overall, I would say like low efficiency is actually favored sometimes too. It's actually a feature for our cloning in this case, right? Because if transformations are super efficient, then every cell would take up multiple plasmids and you wouldn't be able to get isolated clean colonies. So the inefficiency actually means that each colony will almost always represent one plasmid molecule worth of bacteria. So when we do our single colonies, we can start them from like one set of DNA information, right? Okay, so now let's look at PCR and primer design. So how we amplify the ADH3 region from yeast genomic DNA. So there's some music playing in the background. Yeah, shout out to it. Okay, that's fine, that's cool. So what's in a PCR reaction, right? So 6 things go into every PCR reaction. So you should know them, okay? You don't need to know the super hyper specifics of like roughly how many seconds we spend on each element, or like how long denaturation takes, this and that, but you should know the basics, okay? First of all, the template. This is your DNA source. It's important to note that it doesn't have to be pure, it just has to be there and present. So when we do sequencing for DNA, which we'll do later on, you need a really, really pure DNA sample. For PCR, that's not really the case. You need primers, so you need forward and reverse primers. So these are the things that define the amplicon, right? The thing that you're actually amplifying. And we'll talk about the design in the coming slides. The design of these is actually pretty important, so make sure to pay attention. The building blocks, the DNTPs, these are the things that the PCR will actually use to build these different fragments. Thermostable polymerase, so in our case we'll use TAC. TAC is, I think, pretty ubiquitous with PCR, right? Like, it's essentially the main polymerase that you will always use. And it's just because of the fact that when we go through PCR cycles, it needs to survive very high heats, up to 94 degrees Celsius. So it just needs to be thermostable. Mg2, it's a cofactor, which means that you either have too much or too little. That means the reaction will fail. The buffer is just the thing that is the environment, right? Because remember, all these reactions that we're doing need to be maintained in a biological environment, right? So this is the thing that will maintain pH and ionic strength. Cool. Okay, 1 PCR cycle, 3 different temperatures, okay? And just as a quick thing, like, that's a picture of the PCR program. Again, it's in your like full deck of slides. But yeah, let's break it down. So there's 3 main parts. There's denaturation, there's annealing, and there's extension. So there's roughly 30 cycles of these 3 temperatures. So you just keep on cycling through these 3 steps. So denaturation is actually the thing that splits up the strands, right? So this is why we need a super thermostable polymerase, because we want to go from double strand to single strand, but we don't want to actually denature the whole polymer. So this is where the double strand and DNA strands separate, but we want to make sure that the TAC still survives, right? And a lot of other polymerases denature at that temperature, which means that whatever activity they have is essentially useless, right? Because this is the temperature regime that we're operating in. Annealing is, I think, probably one of the most important parts of PCR, primer design, like everything in general, right? Like, I think this is where people like really like in molecular biology, when they're designing primers, like this is where you fail. And I think it's like kind of like the most important part is PCR. So I'm going to focus on it the most. Okay. So the temperature for these have to be just right. Okay. So the primers need to be designed so that, your temperature will be 3 to 5 degrees below the lowest primer melting temperature. And by that, I mean, when you have primers for PCR, you have a forward primer and you have a reverse primer, right? So let's say your forward primer was 60 degrees Celsius, but your reverse primer was 30 degrees Celsius, right? So we don't average those temperatures. We take the lowest primer TM and we subtract 3 to 5 degrees, and that's the annealing temperature. So this is very important. And then extension. So this is where you have TAC actually adding nucleotides from 5' to 3' to the 3' OH end of the primer. So basically, after 30 cycles, you have about 10 to the 9 copies. PCR is exponential, right? So it's 2 to the n. It tends to, in practice, plateau. But, I mean, all you need to understand is the fact that PCR is an exponential amplification, right? Okay. So this was one formula that was covered within your slides, and I think it's pretty important to call out. So this is the Wallace rule. So this is a really good approximation rule. nowadays we have a lot more precise mathematics to describe how to optimize primer temperature. Excuse me. But this was really state of the art back in the day. Okay, so what is it? So this is, I think, the only PCR formula you should just have memorized, right? This is like something that you should know how to do. So melting temperature is equal to 2 times the number of adenines and thymines plus 4 times the numbers of guanine and cytosine. And you should think for a moment why that kind of makes sense, and why guanine and cytosine is worth more temperature than adenine and thymine, right? Because TM is just describing to you how high, like the melting temperature, right? It's the temperature at which 50% of your sample is melted. And G and C have 3 hydrogen bonds versus adenine and thymine have 2 hydrogen bonds, right? So, you know, it kind of intuitively makes sense that guanine and cytosine would lead to a higher TM, just because of the fact that a stronger connection means that you need more heat, more energy to break, to melt them apart. So essentially GC is harder to melt. More heat means higher TM. Okay, cool. This is a quick little example. Again, I would make sure that you're comfortable with this. I think it's like the only like calculation that you really covered within section. So I think it's pretty reasonable to be tested on it. Okay, so let's talk about polymerase choice, right? So we have TAC and we have PFU. So I think that you should be able to compare these different polymerases. So TAC it's awesome. It's phenomenal. It's from a strain called Thermus aquaticus. From the Thermus, you can kind of realize like, oh, thermostable. Like, you know, it's fast, it's cheap, it's the classic PCR enzyme. It's important to note some downsides. It has no proofreading, which means that it also has a significantly higher error rate. So you should think about, you know, why you might want to use PFU. So PFU is slower. It has proofreading, so it has 3' to 5' exonuclease activity. So it's significantly a lower error rate, but it's also a lot slower. So sometimes when you sequence your clone PCR product and you find a single base mismatch, it's likely due to the fact that you had a TAC error during amplification. And if you switch to PFU, it would reduce this dramatically. In practice, I personally have never had to use PFU. But again, I think it's something important to keep in mind. Okay, primer design, right? So I think these are the 6 critical rules of primer design. So the first thing, we want it to be long enough. I don't think it necessarily needs to be between 18 to 22 base pairs. But the general idea is, for every base pair you get, right? For every base pair that you have 4 choices, right? You have adenine, thymine, guanine, cytosine, right? So when you do a calculation for how many potential base pairs you have, it's 4 raised to the power of 18, right? Which means that you have 7 times 10 to the 10 base pairs. So you can adjust the length based on how long your genome is. The general idea is you want it to be long enough such that you don't randomly chance upon a certain primer, right? Like the odds of finding an 18-mer that's not what you were targeting in a genome that's that long are very, very low, right? And that's just a probability thing. 2, GC content should be 40 to 60%. So it's important to note that you don't want to make your GC content too much, because raising the TM too high makes it introduces a lot of issues, especially with things, considerations of secondary structure. So it's important to balance that out. You also want to make sure that it's not too low, because if it's too low, then you have unstable annealing, which means that you get a lot of different products that you don't want. So a TM in the range of 50 to 70 is pretty optimal most of the time. And again, there's variation from that, but I think generally speaking, that's how it goes. For 4, no hairpins or dimers. So you don't want to have self-complementary strands, because you can get secondary structure formation, which becomes really troublesome, because then they'll show up as these low molecular weight bands on the gel as primer dimers, and these different complexes, which doesn't really give you a very good representation of what you're seeing. And then rules 5 and 6 are, again, pretty important. So again, it's important to note that you need to engineer restriction sites on the 5' ends. Actual annealing will happen with the gene-specific 3' part, and then the 5' restriction site will dangle until the cut. That's basically how we add Sal I to the forward primer and BAM high to the reverse for the ADH3 cloning. And then always go from 5' to 3', okay? So this is very, very important. It's an important distinction between a forward and reverse primer, right? So if you're designing a forward primer for a genome, right, that forward primer is going to follow the strand like in the genome exactly, right? So if you wanted to design a forward primer for a genome, like that would be like identical to the gene strand itself. However, when you're trying to design a reverse primer, it needs to be reverse complementary to the end of that genome. So always make sure you're going from 5' to 3'. It's a very important distinction. And if that doesn't really make sense, maybe look at a genome and kind of try to think about how replication works and how you get a forward primer and where exactly it's attaching to and where exactly a reverse primer would be attaching to. Okay, awesome. So now let's look at nested PCR for specificity. So nested PCR is basically you make a rough cut and then you make a precise cut, right? So if you're trying to find a specific paragraph in a book, for example, right, it might be helpful to first identify the page that that paragraph is on, and then after that make cuts to identify the paragraph itself, right? So the idea of round one is you have outer primers, you have a really big amplicon. The amplicon is the region that's amplified in PCR, okay? I actually did not even know that terminology until like an hour ago, so very cool stuff. People in biology in general, they just love their terms. But yeah, anyway, all that to say is a lot of stuff will be off target, and that stuff will amplify, and that's okay, right? There'll be regions that flank the area that you actually want, and that's the point of round two. So round two is your inner primers will anneal inside the round one product. So even if the round one had like off target shit, that shit is like very unlikely to contain the inner primer sites, so it doesn't get amplified. So the overall result is a very clean, nice, specified product. So you'll notice how we did nested PCR here. So that's something that Dylan actually changed. So Dylan has been working with Nikki on this course for a very long time. And in previous years, they tried to do one-shot PCR, but they found that it would often never really work. But we've had very good success with our nested PCRs this quarter. And it makes sense, right? Because the yeast genome is really, really big. It's 12 megabytes of information, which means that non-specific products are very high potential, right? If we go back to that calculation of like how long our primers are, how long a genome is, you'll find that 12 MB like genome is really, really big, and it's actually probabilistically possible that we would get a random site that would map to our primer. But by doing double cuts, we're able to kind of get a second round of primers that are the ones that have the BAM high and the Sal I sites engineered in. And that's the product that we want to clone. It's pretty clever, actually. It's kind of cool. Okay, gel electrophoresis essentials. So we run so many gels in this class, okay? And I think it's really important to understand what we're doing. There's 4 main things that go into a gel. For one, charge is probably the most important thing. The fact that DNA is negatively charged because of the phosphate backbone means that you migrate from the negative electrode to the positive one. So the negative one is the black electrode on our setup, and the red one is the positive. So the black is where we put the back of the gel, and negative repels negative, right? Fragments are separated by the log of the molecular weight. So it's important that you note that it's not a linear relationship, it's a log linear relationship. Small fragments will travel farther per unit time, and it's really good that it's a log linear relationship because you actually are able to see very good separation, even when the fragments are close together, right? Which becomes very important in some cases. For the purposes of our class, you kind of like, it doesn't really matter, but it's useful. Like most of the fragments that we've looked at thus far have been in 1028 base pairs, but yeah, it's important to know. Okay, I think this is important too. Generally, you want a lower percent of agarose for bigger fragments. That means like think of like the agarose as like mesh, right? and lower percent means more open mesh. So 1% also just means 1 gram per 100 milliliters for calculational purposes. And then for an X% gel of V, you can weigh X over 100 times V grams. I think this is a really important formula, and I think like I've not really talked a lot about the calculations. We kind of just take for granted that in our lab manuals, the numbers and the amounts that you want are given to us. But it's really important you understand how we got those numbers and why we're using specific percentages. Another thing to watch out for, I don't think this is really too important for the purposes of this class. Just that the linear ladder is not the same thing as supercoiled plasmid. So the ladder is linear, and you can't size supercoiled plasmids against it. So you just got to keep that in mind. So supercoiled means you have secondary structure stuff. So the idea is how earlier we were talking about how you don't want secondary structure elements. You don't want reverse you don't want complementary things that can bind within each other and produce secondary structure within a single strand or even a double strand. So a lot of what we try to do in this class is we try to linearize it and then we run the gel, right? Okay, cool. Okay, so now we're going to talk about restriction enzymes and ligation. So cutting, joining, and all the logic around BAM high and SAL I. So the basics of the restriction enzymes, right? So there's endonucleases. These are the things that cleave the phosphodiester backbones at specific sequences. They naturally are occurring in bacteria and archaea. They defend against invading phage. So it's important to note that these endonucleases are not something that we've not come up with them. These things exist in nature, but we can engineer specific endonucleases for specific use cases. And that's pretty foundational in the context of how CRISPR was developed as a whole, right? Okay, so the whole point, restriction enzymes are bacterial defense proteins. We co-opt them to make precise cuts in DNA in vitro, right? Bacteria uses them to chop up phage DNA. They can also methylate their own genome at the same sequences so they don't self-destruct. The restriction in restriction enzyme just refers to restricting viral infection from a native standpoint. Type 2 is the most common. So these are the things that cut at palindromes. Type 2S are more unusual ones that cut outside their site. So this is pretty relevant for CRISPR-related techniques, something called Golden Gate cloning. Those are kind of things that you would do if you went further into molecular biology. We'll do CRISPR. I don't think we do Golden Gate cloning within this class, but maybe if you worked in a microbiology lab or in a DNA lab, you've done it before. Enzymes differ in many ways. Some include specific buffer requirements. It's important that we consider this, and that's actually why we're using a double digest sometimes. Sorry, misspoke. Sometimes a double digest will use a compromised buffer. So what this means is a double digest means you're using 2 enzymes, and these 2 enzymes natively operate at different optimal conditions, right? So compromised buffer is just, they see how they can optimize the activity of 2 enzymes while keeping the environmental conditions the same. Cool. So I think there's a pretty important concept here. So I think geometry is the most important part when it comes to looking at primers and looking at enzymes and cuts and a lot of things in molecular biology. So let's look at these different enzymes. So EcoRI, BAM-Hi, SAL-I are 3 different types. PSTI, CFOI are 2 other types. Protruding and sticky is something that we're going to take a really, really big advantage of, okay? So 5' protruding sticky is the most common. So what this means is you get an overhang that sticks out at the 5' end. 3' protruding sticky is slightly less common. You get an overhang at the 3' end. Personally, I've used pretty much all of these. The ones that you very commonly use are all of the 5' protruding ones, and you often use PSTI. Blunt means no overhang, which means it can ligate to anything blunt, which is pretty inefficient. So the critical concept is 2 sticky ends can only reanneal if their overhangs are complementary, okay? So if you have 2 BAM high ends, for example, you can have both having GATC overhangs. However, we cut with BAM high and Sal I because of the fact that these are not complementary, right? So BAM high has GATCC, and Sal I has TCGAC, right? So one of the things that we try to do in this class when we use BAM high and Sal I is to linearize our plasmid, right? So it's important that we use a sticky method of cutting which isn't complementary, because if it was complementary, or if we only used one of them, then it would just religate and recircularize, which is not useful, because if it recircularizes, then we're not going to be able to insert anything into it, and it kind of messes up our experiment, right? So I think this is very, very important. It's at the heart of the logic behind our ADH3 cloning, the fact that we use BAM high and Sal I, specifically so the vector's two ends cannot reanneal to one another. Cool. So I think it's also important to have kind of an intuition around the numbers. You don't really need to know the specifics, but you should have a general idea, right? So we kind of talked about this earlier, but we're going to go through it again. Each base position has 4 possible nucleotides. So multiple cloning sites tend to use 6-cutter and 8-cutters to get unique sites. A 6-cutter means that you have, if you're trying to randomly find a sequence of 6, the odds are 1 over 4,096 base pairs, right? If you try to use a 4 base pair site, the probability of match is 1 over 256, which is actually pretty reasonably high, which is why we want to either use 6-cutter or 8-cutters, just because of the fact that we have a much better likelihood of getting specificity. If we wanted to apply this to a yeast genome, for example, right? 12 megabytes means 12 million base pairs. That means that a 4-cutter would give you 47,000 fragments, which is just way too many, right? Versus a 6-cutter would give you 180 fragments, and an 8-cutter would give you a 6-cutter would give you 3,000 fragments, and an 8-cutter would give you 180, which is more manageable. And this is also why multiple cloning sites use longer recognition sites, because you want exactly 1 cut per plasmid for each enzyme. Okay, cool. Let's talk a little bit about ligation. So we use something called T4 DNA ligase. T4 ligase forms phosphodiester bonds. It needs 3 things. It needs a free 3' OH, a free 5' phosphate, and ATP. So in terms of sticky versus blunt, sticky ends are a lot better because the overhang's base pair, which means that holding the fragments next to each other while the ligase finds them, which means like you're holding the fragments next to each other while the ligase finds them. Blunt also works, but it's a little bit slower, and it tends to also give a much lower yield, which is why we don't like to use it. Some enzymes leave non-cohesive overhangs, so different sequences, which means they don't ligate to each other. This is a trick that, again, we exploit, which I talked about earlier. In terms of procedural considerations, T4 optimally works at 37 degrees Celsius, but we can run ligations at room temperature of 4 degrees Celsius because we want the short overhangs to stay base paired long enough to ligate, and higher temperatures would melt them. So again, this is a consideration of, sometimes you'll have mismatches between what you're looking for in terms of the optimal environmental conditions of your desired outcome versus, the optimal environmental conditions for the use case of the enzyme. So it's important that you make these small distinctions. I think molecular biology in general is reasonably straightforward, except for a couple of nuances here and there that really make all the difference. Okay, well, I guess we're getting the music back. Fire. Okay, so let's talk about the type of cloning that we do. So we talked a lot about blue and white screening, but we don't actually do that. We do something called directional cloning. So this is the most important concept, I think, in the context of BAM high and Sal I. So let's look at this step by step, right? So cutting with BAM high and Sal I, we cut the vector with both enzymes, and the resulting linear vector has 2 different ends, right? BAM high end has a GATC overhang, Sal I end has a TCGA overhang. They're not complementary, so the linear vector cannot close back up on itself. The PCR product also has BAM high on one side because we put it on the reverse primer, and Sal I on the other, which is the forward primer, which means it can only ligate in one way. The vector-only control plate should have 0 colonies. So let's look at the bad case. Let's say we forgot one of these enzymes, right? Let's say you forgot to add one of them. That means that you would have the cut being initiated by the enzyme on both of them, right? The BAM high would cut on both sides, right? So you'd have two of the same overhangs, which are complementary, which means that it would just circularize and it would just be pointless. Like your cut would be pointless. So this is the diagnostic that we use. Many colonies on the no insert plate means we had a digest that failed. For the circular vector transform cells, you should get a lot of colonies on the no insert plates or no insert control. And then this is what directional cloning is. So the insert goes in only one direction because the two ends are different. Cool. So I think we'll cover the calculations in a different video. So this should wrap up all the most conceptual parts. I also want to note that this is in no way like a full thing. Like this is like the highlights of what we've been learning, and I think it's very important that you note that anything on the slides or within the lab manual for the first few weeks is pretty reasonable, fair game. Again, I wouldn't I wouldn't worry yourself too much about we added 750 milliliters or 750 microliters of M1Z plasmid or M1Z buffer and what the components of that are, right? I think if you understand conceptually what's going on, why we made the choices that we did instead of the specific what's, I think you can do very well on this exam.